Good morning, everybody. Welcome to the TRI Cancer Seminar Series. Today we're having the Cancer Imaging Mini Symposium. Firstly, I'd like to acknowledge the traditional custodians on the land we are on today, and we recognise their continuing connection to land, waters and community. We pay our deep respect to them and their cultures and to elders, past, present and future. So today we've got a lineup of EMCRs that are going to tell us about their cancer imaging work, which is trying to increase our knowledge of the cancer disease and also the therapies that we can use against this disease. So our first speaker is Dr. Robert Ju. He completed his postdoc here at UQDI with Professor Nicholas Haas and Samantha Stebbins, and he now works over at the AIBN and IMB with Dr. Stebbins. Thank you, Rob.
Thanks, Shannon. So today I just want to take the time and talk a little bit about a project that's been ongoing for about 7 odd years, finally come to completion now. And I just want to preface this talk by saying that it started in the lab of Nicholas Haas, where I started my PhD, and he was one of my co-supervisors. He's been quite instrumental, as well as my current boss, Sam Stebbins, in driving this project forward and sort of mentoring me through. And the talk is really about how different components of the cytoskeleton, especially microtubules, enable cells to migrate through confined spaces. And this is particularly relevant in the sort of setting of cancer. So this here is a tissue biopsy of a metastatic melanoma, and it's from pulmonary tissue and stained with hematoxylin and eosin. And what you can see here in the purple are all the nuclei of the abnormal and normal tissue. And in the dark purple, you can see a marker of melanoma. And what I hope you can appreciate from this is how packed this environment is, both for normal cells and also abnormal cells. However, studying how these cancer cells got here is quite difficult. This is a snapshot in time. It's a very detailed snapshot, but it doesn't permit us to look about the spatial dynamics and also the temporal dynamics that occur as cells invade through tissues. And that's what our lab does. So we use live cell microscopy and combined with 3D models to study how cancer cells invade and survive mechanically confined forces from confining microenvironments, as you can see by this fluorescently labeled melanoma cell with actin in orange and collagen fibrils in cyan as it tries to squeeze, and it does so successfully through a couple of these collagen fibers. But before I get into the work, I thought I'd take a bit of a historical tangent and talk about the origins of live cell microscopy. And it originated in the US by this researcher called Shinya Inoue. And in the 1940s, just after World War II, he was one of the sort of pioneers that built one of the first polarized light microscopes, and using just a couple scrap parts, as you can see on the bottom left here. So he used a discarded machine gun base, a TCAN tin, a mercury lamp, and a polarizing prism to basically capture this, which is mitosis of a pollen mother cell of an Easter lily. And through very clever optical physics, what he's been able to do is visualize in white these fibers that he describes as being instrumental for cellular division, or as we know it, mitosis. So he goes on to sketch out these fibers in quite explicit detail. And we now know these as microtubules. And we have more advanced techniques to image microtubules now, as you can see here in this melanoma cell. with endogenously tagged microtubules with enhanced green fluorescent protein as it undergoes cell division. And I also think it's quite interesting, quite insightful that in the 1960s, when Shinya Inoue and Sato published this review, they said, when referring to microtubules, we hypothesize that microtubules are a plausible mechanism not only for the mitotic movement of chromosomes, but for movements of organelles and mechanical modulations of the cell surface. So this was at a time where technology was, very different, and his insight has proven correct in the story that I'll tell. So microtubules are these hollow tubes, and they're composed of alpha and beta subunits. And these alpha and beta subunits form protofilaments. So there's 13 of them that form this sort of spiral tube. And within cells, they undergo sort of dynamic growth, shrinkage, and stability. And what you can see in the cyan of this micrograph is individual microtubules within a melanoma cells growing and shrinking over time. And this sort of behavior of dynamic instability at the longer time scale allows cells to evoke their biological and cellular functions, like cell motility here. But microtubules by themselves are actually very fragile. They're stiff and prone to breaking. And this is where this protein, or this family of proteins called microtubule-associated proteins, come in. And this family of proteins is responsible for tuning mechanically and the stability of microtubules to confer very different dynamic biological processes. So CLASP, the protein that we're interested in, is a microtubule plus N or plus tip protein. So it associates at the very fast growing end of microtubules, as you can see on the top left in cyan, by the master regulator EB1. And CLASP just below it, sort of annotated in white, exhibits very similar dynamics to EB1. And that's because it utilizes EB1 interaction to recruit it to the plus end of microtubules. And through its various different domains, so it's got 4 distinct structured domains, these different domains execute different functions towards microtubules. The classical functions that have been described in the past 20-odd years have described CLASP as a potent regulator of microtubule depolymerization. It's responsible for delivering cargoes such as matrix proteins to the cell surface. It's also responsible for organizing organelles and anchoring them, and like I showed before, in mitosis. But the quite interesting thing that happened just after I started my PhD was these 2 back-to-back papers by the Akmanova lab that showed using purified in vitro experiments of microtubules, they could grow microtubules decorated with CLASP, and it allowed these microtubules to enable them to undergo buckling without breakage. So it reduces the flexural rigidity of them, allowing them different mechanical properties. And then they also showed that when you damage microtubules, CLASP gets pretty quickly recruited to these damaged sites to repair them. So it's a really potent protein that modulates the behavior of microtubules. And so this finding was really curious to us because we wondered what the cellular consequences for CLASP, especially in invasive cancer cells, was. And so the first thing that we did was we used this sort of classical assay that we do in the Haas lab, which is we grow spheroids. And these are sort of quadrants, I guess, of melanoma spheroids. And you can see the outline in purple. And when I play the movie, what you'll see is in the control, these cells will invade into the surrounding collagen 3D hydrogels over the time period of about 20 hours. Now, when we knock down CLASP, we get pretty dramatic perturbation of this invasion into the 3D collagen. And we can really nicely quantitate this, but this really doesn't tell us anything about the role of class, apart from that it's probably important in 3D cell invasion for melanoma cells. And so then we wondered, really, what is the mechanism and why does class knockdown perturb how cells invade in 3D? And so what we did was we embedded single cells, and these are cells with magenta labeled with their nuclei in 2.5 mg per mil of collagen. And the collagen below is the sort of the structure, the architecture of the 3D surrounding hydrogels. And in the 37-degree polymerized hydrogels, the cells basically don't move too much at all. They're quite static. They try to sort of extend protrusions and they largely fail. But when we modulate the organization and the structure of the collagen, what you can see is that if I replay this movie, as a cell attempts to migrate through slightly larger pores between the fibrils, the cell basically dies in attempts to do so. And this is because we sort of expand the geometries of the environment, allowing the cells a little bit more flexibility in trying to invade. But actually capturing this is really difficult. And the reason because it's so difficult is because the pore size or the pore diameter of collagen is very, very heterogeneous. And it's demonstrated here in this paper in 2019, where the authors have quantitated the pore size by using spherical filling modeling and quantitated the diameter of these spherical balls within the gaps of these collagen fibers. And roughly in a sort of 3 mg per mil collagen lattice set at 37 degrees, the pore size is about 4.5 microns in diameter. So that's the size that the cells need to actually fit through to basically undergo invasion. Now, this is important for melanoma cells because melanoma cells can actually adapt, as you can see from the last two movies, to their different environmental geometries. And this is here, what I'm going to show you is this concept of mechanoreciprocity, which is the bidirectional relationship between cells and the mechanical sort of properties of their substrates and environments. So these are melanoma cells, endogenously tagged with their microtubules with GFP, and they're grown or embedded in three different settings. So the one on the left is deep within a collagen lattice, a fibrillar lattice. And what you can see the cell doing is it undergoes this predominantly bleb-based motility that we call amoeboid blebbing. So it's a very efficient, fast blebbing motility that uses hydrostatic pressure, local hydrostatic pressure gradients to basically progress motility. And so the classical one that many of you may or may not have seen is this one where cells undergo mesenchymal motility, where they extend a primary pseudopod and they use proteolytic sort of degradation mechanisms to sort of gore out holes within the 3D hydrogels to progress motility. And the last one is the classical one, the sort of the 2D migration, where cells are plated onto very stiff glass. And what you can see here is that in these three different settings, especially 2D versus 3D, is that the microtubules organize themselves in very, very different ways within the cells. And we think this is actually quite important because of the sort of the mechanics of these 3D sort of geometries of collagen hydrogels. And like I said, it's actually really difficult to capture these. So these two sort of videos were probably the only two videos that I was able to capture successfully in my PhD. And so sort of knowing this, we developed this model that recapitulates this premise of confined migration. And the way we do this is we microfabricate these large and smaller circular structures out of silicon. And on the left, what you can see in the highlighted orange, the purple, is a melanoma cell undergoing confined migration between two of these pillars. And a lot of what I will show subsequently will be using this confined model of motility. that we've developed. So when we put our melanoma cells with microtubules in white and nuclei labeled in magenta, what you can see is that they're able to pretty successfully navigate these confinements that we create in front of them. And they do it over the time span of sort of hours, roughly between 2 to sort of 6 hours it takes them to go through. And quite nicely, what we were able to show is that when we knocked down class, we were able to recapitulate this finding that we did in 3D collagen hydrogels, which is as cells attempt to migrate through these confinements, these mechanical confinements, they die. And we know this is geometry dependent because if we allow these cells and relax the geometries and grow them and allow them to migrate in 5 by 5 micron sort of long rectangles, class depletion has almost negligible sort of effect on their viability. So it reduces a little bit of their viability, but they're still largely able to move through successfully without dying, as you can see here. So the question is, what exactly is class doing to allow cells to survive within these confined settings? And so I think the best thing to really do is to show you what the microtubules look like when we deplete clasps from cells as they attempt to migrate through these confined microchannels. So in the controlled cells, you'll see as this cell tries to push its nucleus through, the microtubules bend and deform to the pillars, and they largely remain quite stable as the cell pushes its cell body through. But as this cell on the right with class depleted does so, you can see the microtubules are not nearly as stable as the controls. And we can quantify this using a neat little trick called window sampling, where we draw windows and extract the values and plot how these values change over time. And in these graphs, you can see that in the control cells, not only are the microtubules more stable, but they actually are reinforced. So the fold change in intensity goes up over time, whereas the ones with class depletion quickly depolymerize. So the other thing that sort of we notice with these cells is that when you deplete class, they have quite a lot of difficulty trying to get through, which sort of points out to potentially the inability for them to generate force in the right areas. And so then we looked at potentially the roles of how microtubules and clasps could regulate force activation and generation within cells. And so there's this well-known sort of protein called GEF, which is a nucleotide exchange factor. And the way it works is that it sits on microtubules. And when it sits on microtubules, it is held in its inactive form. And only when it's released from microtubules, from microtubule depolymerization, does it then become active and then activate downstream signaling via ROA ROC and what you and I may know as the classical force generation machinery in the cell, which is actin-myosin contractility. And we initially hypothesized that class was the sort of the main determinant of how GEF-H1 could activate in these settings. So basically the way class works would be just to stabilize the microtubules, thereby preventing GEF-H1 from activating and then triggering downstream actin-myosin contractility. So when we look at what class does in these settings, what you'll notice is that there is this sort of differential gradient of how class is in terms of its stability. And what we see is that at the rear of the cell, at the very top, class stably binds to these sort of lattice structures, which are microtubules. And then progressively towards the front, it becomes more and more dynamic, suggesting that it's preferentially binding and stabilizing microtubules at the rear and triggering some sort of polarized force activation. And so when we look at all this sort of downstream signaling using biosensors, we can see that indeed GEF-H1 does also activate at the rear. And quite nicely as well, myosin through actin-myosin contractility is also enriched at the rear. And we think that this is principally driven by clasp localization and stabilization of microtubules at the rear, enabling cells to generate this contractile force at the rear in order to push the nucleus through. Now, what happens when you deplete clasp is that you get complete dysregulation of this process. You no longer get GEF-H1 recruitment or enrichment at the rear. And you can see here as GEF-H1 activates, it slips completely past and then it doesn't enrich and the cell dies. And the same thing as well with actin-myosin contractility. We just get complete dysregulation and activation of this force machinery in the cells. So from this sort of body of, I guess, experiments, we were able to show quite nicely that clasp actually stabilizes microtubules, but in doing so, controls the force-generating machinery in cells. But we actually haven't shown so far, or I haven't shown so far, that clasp-decorated microtubules are indeed more mechanically resistant to forces. And this is actually quite difficult to show. And this is why we've sort of changed the method that we use now to test how cells respond to force. And so this is the system that we use, and it's basically a system that allows us to squash cells. And so on the top left, you can see this is a CAD drawing of the mold. So it's these concentric circles machined out of aluminium. And on the bottom, that's what it looks like after it's completely assembled. And then we pour in silicon and then demold it, and it becomes like this suction cup. And when we attach tubing and hosing to it, and then attach it to this syringe with a liposuction syringe lock, what it allows us to do is deploy the piston down. And what you can hopefully see on this image, sort of two from the left, is that there are little circles on the flat surface of the device. And this actually is little pillars that we microfabricate into distinct heights to control how much we confine or compress the cells. And so what you can see here is that in 3 different cells, unconfined, and then we've sort of fabricated some 3.7 micron pillars and some 11 micron pillars, and we can actually compress them at different sort of degrees and basically look at how different components of the sort of microtubule cytoskeleton behave in response to direct force. So when we basically do this experiment and grow cells expressing tagged microtubules and the nuclei in black and magenta respectively, and we compress them, what you can see here on the left is that the control cells actually exhibit microtubules that are vastly more stable than those of their class 1 depleted counterparts. And this has a lot of different outcomes towards cells. So when we add in a dye called propidium iodide that basically crosses the membrane once it gets breached, we can see that when we compress class depleted cells, we get dramatically more PI influx. So there's things getting into the cells indicating there's membrane rupture occurring. The nucleus size also increases quite a bit, suggesting that the absence of these stabilized microtubules allows cells to feel more force and then expand in response to force. And then finally, their microtubules are pretty significantly more fragile. So in response to compression, the microtubules basically depolymerize, and then the cells obviously die. So this is sort of a direct experiment that shows if you don't have clasp stabilized microtubules, they're not as resilient to force, and cells suffer as a consequence of this. And we think that this plays quite an important role in how cells navigate these confined pillars that I showed before. So now what exactly is class doing to sort of evoke this, this function? So what we've done here is we've generated some endogenously tagged class. So we've used 2 GFPs and we've knocked it into the loci of class to visualize endogenous clasp behavior. And what you can see here is that, as it should, it grows on the fast growing ends of microtubules. But I think this is probably the most exciting experiment that I've done in my life, I guess, which is when you compress clasp endogenous stable cells, you can actually see that it changes in its sort of stable loading. So it basically now loads onto microtubules directly. And this process is actually reversible. When we remove the confinement, you see this reversion. So this suggests that clasp is actually directly responding to mechanical force to stabilize the microtubules, giving cells more resilience to force. Now, so what is the sort of the exact molecular mechanism that clasp does all of this through? So this here, this sort of blue and orange is basically an AlphaFold prediction of what clasp protein looks like. And what you can see in the blue, which is a high confidence prediction, is the 4 very structured domains of clasp. And it's separated by this intrinsically disordered region, or this very flexible sort of peptide region. And we think that it basically expands and allows all these domains to interact with different conformations of the microtubule lattice, thereby stabilizing it. And so there's a lot of literature that suggests that the first 2 TOG domains are actually the ones that confer mechanical resilience to cells or to microtubules. And so this is what we've done. We've expressed half of the protein and the other half in different cells and watched where it goes. And quite unremarkably, the TOG1/2 actually doesn't do much at all. It just exists in the cytoplasm. Whereas the sort of the C-terminal fragment of clasp quite nicely localizes to the growing plus ends. And this is known because the C-terminal domain contains a sequence that recruits it to the plus ends. But what's actually really, really nice that we can show is that when we compress these cells, you can actually induce the loading of clasp, just the TOG1/2 onto microtubules. And it occurs within about 15 minutes or so. So sort of armed with this knowledge, what we've done is we've utilized an optogenetics approach. And just for the lack of time, I'll just quickly go through this. And we can insert an optogenetic module in between proteins to separate them. And you can do this within almost any protein. And upon exposure to blue light, you basically dissociate 2 halves of the protein. And along with Torsten and Jeffrey, we were able to do this in our melanoma cells using a construct that Jeffrey had generated previously called PyEV1. And it allows us to dissociate the growing plus ends from microtubules in a subcellular sort of fashion. And so we applied the same approach to our clasp, and we divided clasp into 2 using this optogenetic module with the idea that upon blue light exposure, we could dissociate this sort of the mechanical conferring abilities of the TOG1/2 from the C-terminal domain. And after probably about 6 months worth of work, we were able to show that we could actually do this. So this is in 2D cells. We can actually dissociate the TOG1/2 from where it's supposed to be. And I guess the functional validation of this is showing how this responds, or how cells respond in 3D. So on the left, you can see this is cells expressing the pi class with no blue light illumination. And basically, they're embedded in a 3D collagen, and they can quite nicely invade over the sort of time of about 20 hours. And when we expose them to blue light, even though there is still a little bit of motility, we can really drastically reduce how much they can move within these confined settings just by dissociating the 2 sort of halves of the protein. So in summary, so what I've shown today is using principally quantitative live cell microscopy and also molecular manipulation approaches. We've identified a role that clasp microtubule interaction plays during confined migration. And then this mechanism is a microtubule dependent mechanism that allows cells to both resist and activate forces. And I'll finish with a couple sort of outstanding questions that we have. And things that we're working towards, which is trying to understand the molecular reason as to why TOG2 actually binds to microtubules when cells are compressed, and also whether this process or mechanism is also extendable into different cancer types, which will basically allow us to target it and allow us to sensitize cancer cells to force as they invade. So just to finish up, I want to thank all the people that have played their part, big and small, and all the grant funding that's enabled this work. And I'm also happy to take questions.
Wonderful talk, Rob. Have we got any questions? No? I've got one.
Yeah, go ahead.
With you're looking at melanoma there, and there's a certain class that's upregulated. Have we looked in other cancer cells to see if it's common? Are they upregulated, sorry, or are they just recruited in response to the pressure?
So I haven't looked at how class behaves in other cell types. I have had a look at RNA-seq data to look at the generalized expression of class in lots of different cancer types, and it's sort of ubiquitously expressed. Class 1, the paralog anyway, is expressed in a lot of different solid cancers and also, blood cancers as well. So it suggests that it could be a mechanism where cancer cells use to resist and activate forces.
Yep. Got a question?
For immune cells that go through extravasation through the blood barrier, is it the similar processes? And are clefts moving in response to bending of the microtubules, or are there other sensors that direct clefts to the microtubules?
So I guess the first part of your question is, do the immune cells use this? So my answer to that is no. Immune cells use a very different mechanism. They do use microtubules, but when you use like a drug like nicotazole to ablate all the microtubules in immune cells, they can still very effectively squeeze through small spaces pretty quickly. And the second part of your question was, sorry?
Clefts to the microtubules, is it response to the bending of microtubules or some other mechanical response?
Yeah, so this is, I guess, a really good question. We don't know. We sort of hypothesize it potentially could be due to breakages in the microtubule lattice. It could be, bending. It could be splaying, and the different TOG domains could potentially allow class to recognize those damaged forms that allow it to recruit there. So yeah, it's a good question.
Hi.
Last question up there.
Yeah, just thanks. From memory, class has plays a role in focal adhesions, and I just wondered, is your mechanism linked to focal adhesions?
Yeah, so that's actually a really good question. Initially, actually, the whole idea of my PhD project was to look at how class targeted focal adhesions to deliver MMPs to enable cells to sort of undergo proteolytic forms of movement. But these cells actually don't use too many focal adhesions. So this appears to be a focal adhesion independent mechanism, especially with melanoma cells anyway. So I haven't looked at, focal adhesion dependence, but in our case, it's not. I don't think they're either, they're sort of in between, really. I don't think, they don't really exhibit any of the sort of the classical features of epithelial or mesenchymal. I mean, proofs in the pudding, like they can even do amoeboid migration. So they're sort of this potluck of cell types, really.
Thank you for the nice presentation. My question is, when you mimic the microenvironment, is it the density or the pressure similar to the physiologic microenvironment, for example, the stroma, or is it hypothetical density?
So your question is asking about the PDMS confinement model?
Yeah.
Yeah. So it's basically a model that we created that induces quite a lot of cellular stress that has been shown to occur during 3D cell invasion. So things that I didn't show today is that as the cells go through these confinement channels, they undergo a nuclear rupture and they can efficiently repair this. And then this process actually is used by cancer cells to sort of survive confinement. And so that was the metric that we used to test whether our confinement devices was sufficient in inducing enough mechanical force. So yes, it is similar to the microenvironment and the stroma.
Okay, thank you very much. That was amazing, Rob.
Thanks.
Okay, so our next speaker is Dr. William McGahn. He is a clinician. He's on call, so we're very lucky to have him here. Will is also doing his PhD in the MARTA. So welcome, Will. His talk's on detecting high-grade dysplasia and stage 1 PDAC.
Thanks so much, Shannon. Yeah, so as introduced, I'm Will. I'm a general surgical trainee, and I'm currently doing my PhD in pancreatic cancer, and early detection is certainly a focus of that. My supervisors are actually all clinicians, but I've had the really great opportunity to be able to work in Professor John Hooper's lab and do a little bit of basic science. Certainly nothing on the level that we've just seen, though. So some acknowledgements and no conflicts of interest. So I think most people here, has this got a like? Yeah, most people here would be familiar with this diagram of the progression of low-grade dysplasia to high-grade dysplasia in the pancreatic ductal epithelium and the stages of pancreatic cancer, invasive pancreatic cancer that come after this. If you're not, then you're probably well aware of the fact that anyone who gets to the stage of invasive cancer has a pretty terrible prognosis for this disease. What you might have been less aware of, though, is that cliff in survival actually occurs between stage 1 and stage 2 cancer. And the key difference between the two is the presence of lymph node metastases. But the reason outcomes are still so poor is because stage 1 cancer still only represents about 12% of resected cases. And that's of patients who actually proceed to resection. And we know that from surveillance studies in high-risk cohorts that early malignant lesions frequently evade detection with the modalities that we have. So if we're going to make really any difference when it comes to early detection for this disease, then we need to target this golden, apparently very brief window between high-grade dysplasia and stage 1 pancreatic cancer. Because we know that at this stage, progression to incurable disease is inevitable. But if we can remove it at this stage, then it is a curable disease. So gallium-68 labeled fibroblast activation protein inhibitor, FAPI. It's a novel radiotracer which targets fibroblast activation protein, which is a cell surface receptor, which gets its name because it's expressed on the cancer-associated fibroblasts, that is the malignant tumor stroma, which I'm sure everyone in this room is familiar with. But it's also somewhat of a misnomer because FAP is also expressed on the epithelial cancer cells themselves. Our hypothesis is that because FAPI-PET is proving to be very effective at finding distant metastases that weren't otherwise known about, maybe it too can find very early primary tumours that other modalities can't see. Only one study has really looked at expression of FAP in the tumorigenesis of pancreatic cancer. That was out of Heidelberg. And what they showed was that invasive PDAC has higher stromal FAP expression. They didn't look at the epithelial cells than low-grade dysplasia. But what we don't know is whether FAP expression can make that granular distinction between high-grade dysplasia and low-grade dysplasia, or whether or not it can even detect stage 1 pancreatic cancer. It was not part of the analysis. And crucially, no one's looked at expression on the epithelial cells in tumorigenesis either. So what we did is we took biobanked cases from the Australian Pancreatic Cancer Genome Initiative. We took 48 cases of either biopsied or resected chronic pancreatitis, because we know that pancreatitis, like other PET traces, is a false positive on FAPI-PET. So we wanted to understand more about that. We had 13 cases of resected intraductal papillary mucinous neoplasm, that's an IPMN, with low-grade dysplasia. Now, for those who don't know, an IPMN is a precursor to pancreatic cancer. It is a neoplasm of the ductal epithelium, but what is unique about it is that it's macroscopic. You can see it. And if you can see it, you can resect it, and that's why we have specimens containing IPMN. It represents about 15% of eventual pancreatic cancer, but it's so important because it's our only opportunity to study, well, not only, but our best opportunity to study this disease in its earliest stages. So we had 13 cases of IPMN with low-grade dysplasia, 11 cases of IPMN with high-grade dysplasia, 21 of stage 1 PDAC, and we had, unsurprisingly, quite a few more stage 2 to 4 PDACs that were available to us. We performed immunohistochemistry against FAP on tumour microarrays of these specimens. And we scored expression not only on the stromal cells, but on the epithelial cells in these lesions as well. And this is what we found. First and foremost, overall FAP expression, that is the sum of stromal and epithelial expression, becomes overexpressed at the point of high-grade dysplasia and remains elevated through all stages of PDAC. Epithelial expression peaks with high-grade dysplasia, whereas stromal expression, that's the cancer-associated fibroblasts, increases with increasing malignancy. Looking at it through a slightly different lens, as in the frequency of positive expression, you see a similar story. But what it also highlights is that in these pre-invasive neoplasms, it's actually the epithelial expression rather than the stromal expression, which is more prominent. And it increases in frequency from low-grade dysplasia to high-grade dysplasia. And then in those in those stages of PDAC, you start to see a more either mixed or stromal only picture. And what's also important is chronic pancreatitis. It expressed FAP, but almost never on the epithelial cells, suggesting that this epithelial expression is a unique feature of neoplasia. So we present a new model of FAP expression in the tumorigenesis of PDAC. One where expression actually begins on the evolving dysplastic epithelium and is followed soon after by adjacent stromal expression. So these are some of the immunohistochemistry slides which tell a good story. So this is chronic pancreatitis, and you can see the immune infiltrate up here. These are normal pancreatic ducts, non-neoplastic. And as everyone here, I'm sure, is aware, the brown is the staining for FAP. And you can see that it's only in that fibrous stroma which is surrounding the ducts, which is classic of pancreatitis. This is low-grade dysplasia, in which you can, in this case, there's actually no epithelial FAP expression and no stromal expression. But this is high-grade dysplasia, which is characterized by that nuclear pleomorphism and architectural distortion in the epithelium. The epithelial cells are quite strongly expressing FAP, and adjacent to this, you can also see this stromal expression beginning as well. And this is full-blown PDAC, which is characterized by these invasive epithelial cells surrounded by cords of stroma. In this case, there is quite strong stromal FAP expression, but there is still a moderate amount of epithelial expression. So we didn't just do IHC, we've also so far recruited 6 patients with a cystic neoplasm, so that's someone who we think has an IPMN, to undergo PET scan with gallium-68 labelled FAPI. And we followed these up for 2 years. So 4 out of the 6 patients had focal avidity at their pancreatic cyst. And of these, 3 ended up having malignant transformation. 1 was offered surgery on account of the fact that it appeared that he'd had malignant transformation, but he declined. And the 2 that were not avid and have not shown any evidence of malignant transformation 2 years down the track. What was most interesting, though, was one particular patient who had a FAPI avid cystic neoplasm in the head of her pancreas. And that's what you can see here. And it's a sustained uptake from 10 to 60 minutes. This patient proceeded to a Whipple's procedure. That's removal of the head of the pancreas along with the duodenum. And histology from that showed an IPMN with high-grade dysplasia. And we were lucky enough to do immunohistochemistry against FAP on this against FAP on this specimen. And what you'll see in this case is this really nice membranous epithelial FAP expression. But what's important about this particular case is that when you zoom out, the surrounding stromal reaction actually had very little or virtually no stromal expression. And what that proves is that this epithelial FAP expression, which we think is the earliest sign of malignant transformation, is detectable on its own on PET scan, which was important to us. There are limitations to what we've done here. You know, they're very small cohort of patients, only 6 who've undergone FAP. We've since expanded that cohort with the help of our colleagues at Hurston Imaging Research Facility to about 36, but we need to wait and see what happens to those cysts to really know the outcomes of that. We also don't know what level of FAP expression on IHC correlates with avidity on the scan, and that's something that we hope to establish with our larger cohort. And we couldn't include PAN-N. PAN-N is kind of like IPMN's more sinister, more naughty younger brother. It accounts for 85% of eventual PDAC, but it is invisible to any imaging modality that we have at the current time. And that's why it's so hard to study, because if you can't see it, you can't resect it and you can't do experiments on it. But we did sample a PAN-N, incidentally, in a case of chronic pancreatitis. And these show up frequently. And in this case, you can see, again, that there is this epithelial expression, and there's a stromal reaction developing around this one, which isn't FAPI avid. And this scan can detect lesions as small as 4 millimeters in size. In fact, this was another one of our patients who had pancreatic cancer. The reason for doing this scan was to look for distant metastases. This is about a 5 millimeter peritoneal deposit, which was not visible anywhere else. PAN-N by definition is about 5 millimeters, but we think in more recent literature that the high-grade PAN-Ns can get up to about 1 centimeter. So maybe, just maybe, not only will this scan have a use in determining the malignant potential of IPMNs, but just maybe we'll be able to detect these previously undetectable PAN-N lesions as well, particularly those that are high-grade. Anyway, thank you.
Thank you, Bill. Amazing, going from inside the cell to looking at our actual neoplasms. Questions? Yes?
Yes.
Thank you for this nice presentation and innovation. Is it the expression of the epithelial compartment of FAP a sign of AMT, or if the cell undergoes some sort of AMT, or is there any reason behind it for expression in the epithelial?
You might have to forgive my lack of background in the basic sciences. What's AMP, sorry?
It is a epithelial mesenchymal transition.
Oh, yes. Yeah, absolutely. That is a question that I have. We know that this epithelial to mesenchymal transition is a feature of high-grade dysplasia. It precedes invasion. Whether or not FAPE is a marker of that, we don't know. The role of FAP in health and in disease isn't well understood. As you can, see from this, very few people have even recognized the fact that it's not just fibroblasts which express FAP. And certainly listening to that, the previous talk did make me, wonder about, what is driving this, mesenchymal transition, which eventually leads to invasion. But whether or not FAP is involved in that, I don't know. But what it appears from these results is that it is involved in that epithelial to stromal communication, which we know is a late feature of high-grade dysplasia and then invasive cancer.
And my second question is, have you done alpha-smooth actin as a control for the FAP in your samples?
Say that again, sorry.
The alpha-smooth muscle actin that is usually expressed in the cancer-associated fibroblasts.
So as in a fast-acting...
The alpha-smooth muscle actin that's usually expressed in the cancer-associated fibroblasts. And then it is been believed that it is a marker of cancer-associated fibroblasts.
It is, and obviously we measured expression in the fibroblasts, well, in the stroma, as many others, as previous studies have done. There's no doubt that it is heavily expressed in the stroma. That's always been the focus of FAP, but I think what we've suggested here is that there is, the epithelial expression is relevant too, and that's probably how it will be used to detect pre-invasive lesions, perhaps, in combination with stroma.
Thank you.
That's a quick one.
Oh,
Yeah,
sorry.
thanks a lot.
We've just got one here, and
Yeah.
then we'll just have to cut it off in the.
It's a quick one. With the limitation number 2, that you can't, that you are not sure at this point if you can define a benign versus a malignant lesion. I guess sensitivity is the important thing right now. As for specificity, you have other markers anyway. So can't you do a fine needle biopsy once you have a highly sensitive signal here, and then you go for specificity in the second round?
Yeah, I see what you're saying, and I wish in practice it was that simple. The way of getting a biopsy of the pancreas is not at all straightforward. The standard of care now is that we do an endoscopic ultrasound, and under ultrasound guidance, we put a needle through the wall of the stomach, and if you don't come back with cancer, if that's not if the biopsy doesn't come back confirming cancer, you don't know that you just haven't missed the focus. So this is a very sensitive modality, but I do think the issue with the limitation of FAP is going to be its specificity. It lights up with all sorts of things, and it's up to the current work to try and figure out, well, can we figure out what represents benign versus malignant? Yeah, and that's hard.
Thank you. When you do the PET CT scan, we have seen that you have used 68 gallium.
Yes.
So we have seen that there's 2 time frame. One is 10 minutes, another one is 60 minutes. So my question is that why there's a time difference, one, and over the time there is 68 gallium has been decayed. So is there any effect of the 68 gallium decay on the experiment?
So the half-life of gallium-68, I believe, is about 7 hours. I have to check that. So the loss in signal from 10 minutes to 60 minutes, as we understand it, is more excretion and loss. Not, how do I put this? the tumor is releasing the tracer. I don't, and obviously part of that's going to be, you know, decay of the radioisotope, but I think mostly it's actually just excretion of the tracer over time.
Okay, thank you very much, Will.
Thanks.
Amazing. Okay, our next speaker, big claps. Our next speaker is Jordan Kumar. He is a third-year PhD student in the HAS lab here at the Fraser Institute, and he'll be talking about the spatial assessment of Ki-67 to stratify MITF in primary and metastatic melanoma. Thank you.
Good morning, everyone. So thank you, Shannon, for the introduction. As she said, I'm a PhD student in Nicholas Haas's lab, so we're going back to melanoma now. If you've heard any of Nicholas's talks, we look a lot into a specific protein called microphthalmia-associated transcription factor, or MITF. And as a brief background of why we look at this, we know that it controls cell cycle and homogeneity of the cell cycle itself. So what we do is we look at the Fuji system. So from this diagram here, you can see as cells go through the cell cycle, they start off expressing signal that fluoresces them in red. And then as they move through S phase all the way up until cytokinesis, they become green. And what we get is beautiful images like this. So this is an example of a mouse tumor that's MITF high. And as you can see, there's a homogeneous distribution of both green and red cells throughout the entire tumor. However, in MITF low, what we get is we get clustering of these specific populations. So we have clustering of these rested cells and clustering of the proliferative cells. So this is all stuff that we've found in vitro and in vivo, but something we actually haven't looked at is how this actually works in patients and whether these phenotypes exist in patients. So what we did was we looked into Ki-67. So Ki-67, as many of you might know, is a commonly used marker in melanoma and breast cancers for prognosis, and it's an S-phase marker. So we're going to use this as a proliferative marker in this kind of case. So this would be our GT or green cells in this analysis. And we're looking at how that correlates to MITF expression in patient samples. With the running question of, does MITF expression positively correlate to proliferation, as we see in vitro? So what we did was we got 187 patient primary samples and 22 patient metastases. And we did subsequent sections, and we stained one side with Ki-67, one side with MITF. And we ran the Ki-67 slides through a cluster analysis pipeline that we developed with TissueGnostics in StrataQuest. And we scored or stratified MITF to high and low. And then we tried to draw correlations to see if everything works systematically from patients to, from what we see in the lab to patients. So this is just an example of how the pipeline works. And this is actually available to use for any IHC markers that you want to look at. I believe it's up to about 4 different markers you can look at in IHC that you can analyze at a single time. So this is just an example of a melanoma, patient primary melanoma that's stained with Ki-67. And with the brown cells being the positive cells and a counter stain of hematoxylin for all negative nuclei. So what StrataQuest does is it automatically segments these cells, and we can get a few different metadatas from these, being different sizes, intensity of each marker, and a few different other things that we can correlate. From here, we actually also used intensity and nuclear size to eradicate and remove any of the other infiltrating cells, because we only want to look at melanoma itself. And then from here, we actually are able to draw a network between likewise cells to kind of assess clustering. So from this diagram here, if you look at the red cell here, what we do is we can have a modifiable pixel distance threshold that we can control. So we draw a radius around every single cell like this, and then we draw connections to likewise cells. So if the red cell here was blue, it would draw connections like this. And if it was orange or Ki-67 positive, it would draw connections like this. And these connections are overlaid in an image like this. And this is how we kind of determine clustering and likewise cells nearby each other. From this, we actually enclose these areas encapsulated by these networks, and we measure these areas and denote them as a single cluster. And that area is also spat out to us, and we can use that as metadata as well. So that looks like this. And we did this across a couple of different pixel thresholds to try and see how clusters change or how sharply demarcated these clusters are to differentiate between or correlate between MITF high and low. So this was the understanding that as MITF, it does push and drive proliferation and homogeneity. If we have a KSD7 pattern that's heterogeneous, like on the left here, as we have a large pixel threshold and we enclose it and we measure that area, as we shorten that pixel threshold, we get smaller and smaller areas demarcated, and that area rapidly drops. So what we get is kind of a demarcation like this across the entire tumor. Whereas if you have a homogeneous expression of KSD7 or proliferative cells, you get an even measurement of this area across any pixel threshold because all the cells are directly next to each other anyways. So you get more of an expression like this. And the area that we encapsulate and take from this, we can then plot with green on the right here being the hypothesized to be MITF high because you have that homogeneous spread. So you get a very short decline in that area ratio. Whereas if you have that sharp changes, you get a very drastic drop across the area and the pixel threshold that we use. And essentially what that looks like as you overlay them is this image here. So here we have 5 different pixel thresholds that we tested from 200 pixel distance to 25 pixel distance. And as you can see, I don't know if you can see my marker, sorry. So we have this, the maximum pixel distance encapsulating the majority of the tumor outlined here. And then as we shorten that down, we get these shorter areas in green, then to yellow, then to orange, and then to red. So this is what we did to look at clustering in KSD7, but how do we actually score MITF? So with the MITF staining, we actually had a very difficult time looking at these guys. So on the left here, we have 2 images that we denote as MITF high, and on the right, we have 2 images that denote as MITF low. However, there's quite a bit of overlap in similarity in the look of the top left and the top right. So what we looked at is specifically how MITF works in terms of proliferation and causing homogeneity. So if we take a closer look at these cells, we actually only looked particularly at nuclear expression of MITF. So the purpling of the cells here and here, comparatively to the blue cells here and the tanning effect, which is just cytoplasmic expression of MITF here. And that's how we denoted what's MITF high and what's MITF low. And these scores were correlated across 3 different people, 3 different blind scorers. We came together and then we all agreed on which ones we all agreed on and then progressed forward with them. So what did we find? Looking forward, actually, when we graphed these curves and we normalized them, we measured the hill slope or the drasticness of that change in area, and we actually found a difference in the correct direction that we were looking for. So with low MITF samples samples that we stratified as low MITF, we actually saw a higher hill slope or a more drastic drop in that area encapsulated. Whereas with high MITF, we had a more gradual or a shorter hill slope. And this was only present for tumors of grade 3 and above, so including the metastases. The reason for that was because we believe that in 2D, we actually cannot see the effect of MITF homogeneity and heterogeneity as well. And so we need something larger than a thin melanoma or something that doesn't have a full 3-dimensional structure to see the effect that we're actually of this phenotype. Looking at the metastatic samples, even though we did have quite a few, a lot fewer samples, with the top 5 highest and lowest MITF scored samples, and we've got a similar trend. However, we are lacking a bit of power here purely because of the sample size. Interestingly, we came across a few other hurdles in when looking at these phenotypes, in that MITF itself has intratumoral heterogeneity. So the expression of MITF itself is not consistent throughout the entire tumors. So on the left here, we have the KS67 stain of the exact same tumor on the right, and this is the MITF stain. So as we look at the area encapsulated in red, we actually see that for KS67 positivity overlaps directly with where MITF positivity is, even in this intratumoral heterogeneity state. So this area is completely negative for KSD7 is also negative for MITF. And this also works in a gradient effect as well. So down here, as we look at the left side of this lower image, we see higher density of KSD7, and that drastically drops off into lower density of KSD7, and that correlates to the intensity of MITF drastically dropping off as we move across that gradient. And then when we measured this in both the primary and metastatic melanomas where we saw this phenomenon, we had a very consistent effect with high MITF. You have high density of proliferative cells. And as we moved to through the same tumor into a lower area, we saw a drop off in that density, except for one sample as an outlier. So what we found from the entirety of this is that we actually developed a pipeline able to assess clustering of immunohistochemical markers. And interestingly, the phenotypes that we look at in our lab are actually very real and they do exist in patients. Moving forward, I'm currently actually looking at how MITF interplays into treatment efficacy and those kinds of things, because we also do know there's a lot of things involving the ECM that MITF does control. I think I went a little bit fast, but thank you everyone for listening, and that's it from me.
Thank you, Jordan. That was great.
There's
Oh,
a few more slides. Oh, that shouldn't be there.
let's get from quick.
Yeah, don't worry about the
Extra.
screen. Don't worry about anything that's happening on the screen right
We
now.
might just have one question just to keep us back on time. If there's any? Oh, just a few extra.
Sorry.
That's all right. No? Okay, well, in the interest of time, we might go forward. Thank you. Hands together, please. Okay, so I'd like to introduce our next speaker. We've got Dr. Zhao Tan. Zhao did his PhD in the Nuan lab at IMB, and they've now moved to QAMR, so welcome.
Thanks for the opportunity for inviting me to give a talk today. So I'm Xiao Tan from Genomics and Machine Learning Lab at QMR. So today my talk is mainly based on cell type community detection using spatial proteomics data in melanoma tumor microenvironment. So the skin cancer is the most common cancer in Australia. Almost 2 in 3 Australians will be diagnosed with skin cancer by the age of 17. And there are 3 primary types of skin cancer, BCC, ICC, and melanoma, and each type affects different layer of skin. And melanoma is the most dangerous one due to its potential to spread rapidly. So I'm sure that we are all aware of the complexity of the tumors. So here I just want to highlight the point that tumor heterogeneity both between patient and within one tumor microenvironment is something that really makes the cancer so difficult to understand and treat. So to successfully treat patients and make meaningful discoveries to improve the clinical translation of such complex disease, we really need to analyze those complex structures and cell communities within the tumor environment. So the spatial omics technology is the way that we can access the cell type communities. And in our group, we have a skin atlas project where we collect 12 patient samples and we apply different spatial omics technology. And those technologies can measure like different modalities like RNA or proteins, and the resolution can be single cell or the group of cell, even the region of interest. And for today, I will mainly focus on the phenocycler fusion platform, which measures protein at single cell resolution. So those are the 6 samples that I will analyze during this talk. And those 6 samples are 3 of them are fatal and 3 of them are non-fatal, and they are mixed in 2 experiments. And I will talk about this later. So look at the each individual samples. The codex data can be considered as a multi-channel like images. So for each channel, we can measure 1 proteins along with this dipy staining. So the dipy staining is the staining for nucleus. And here I just show 3 other protein stainings. S100B and PML are common marker for melanoma, and keratin 14 is a marker for epithelial, in this case, keratinocytes. And to get the single cell data, to get the protein data at the single cell resolution, we first need to perform the cell segmentation on a dipy channel, and then we can aggregate all the protein signal from different channel and generate the single cell data. So once we generate the single cell data, we can use the annotation of the markers to annotate each cells. And in this experiment, we measured about 33 proteins, and we can annotate those amount of the cell types. Those cell types can be the real cell types or some cell stage, and also the positive molecular markers. You can see that at the global level, this cell type annotation is quite noisy. So we come up with a way to perform the cell type community or cell type niche analysis, which means a group of cells that interactive with each other in a specific environment or tissue regions. So when we perform this cell type community analysis, we can group those different cell type into like several communities, and each community has a different cell type compositions. So for example, we can see that community 3 and 4 are 2 melanoma communities. And we can see that the cell type compositions in those communities are very different. For example, the community 3, which is in this region, enriched with melanoma and also the fibroblast. And we can also zoom in those regions and annotate each cell community with annotation. And we can also visualize the colocalization between different cell types. So in this case, I'm interested in the melanoma and fibroblast. So we can see that in those regions, those 2 cell types are have high chance to close with each other. And this is quite interesting because normal dermal fibroblasts can inhibit melanoma growth. However, due to the time changes, the fibroblasts can become cancer-associated fibroblasts, which support the melanoma progression and metastasis. We can also visualize the cell type community and cell types in a network graph. And in this case, we can see those, there are 2 melanoma communities, the one associated with the fibroblasts and the macrophages, and another one with other T cell and immune cells. We can apply the similar approach to compare the non-fetal and fetal samples. So in this slide, we have 2 non-fetal samples and 1 fetal samples. And we can see there are 1 community stand out in non-fetal, the non-fetal samples, which is the cluster, the community 3, and enriched with MHC class 2 signals. When we apply a similar approach to another slide, which has 2 fetal sample and 1 non-fetal sample, we can see the cluster 6, which also stand out in non-fetal sample. They also enrich with MHC class 2. And we can also see that in fetal sample, there are unique cell type communities which enriched with blood vessels. And if we consider like each community as a geometric shape, we can then calculate the like the geometric statistics. For example, we can calculate the area of different cell type communities. And in this case, we can see this MHC class 2 community has a larger area in non-fetal sample compared to the fetal sample. And in another slide, we can see that there are more blood vessel in fetal sample compared to non-fetal sample, which may suggest the vascular invasion of the melanoma. We can also find the distance between the closed niches for each cell type community. And in this case, we can see that for the melanoma communities, the closer distance with the fibroblast is shown in this heat map. And we can see in fetal sample, the melanoma are very close to the blood vessel, but for the non-fetal sample, they are far away. It also suggests that there may the vascular invasion may happen in those samples. So in conclusion, we can identify the key cell types in the skin cancer use spatial proteomics data. And our analysis can also classify cell type community and also detect the cell localizations. And we also use the spatial statistics like geometric shape to enhance the understanding of the cell distribution and interactions. And we think that those cell type community can be a new feature to study the cancer progression and prognosis. Yeah, thanks.
Thank you very much. That was awesome. Have we got any questions? I've got one. You found lots of different niches there. How do they compare to what a clinical pathologist sees? Is there any correlation between the different niches? I know they're not seeing as much detail, but
yeah. For this data, we don't have pathologist annotation, but in this study, we also have other data set and we have the pathologist annotation. And the like melanoma, they are correlated quite well with pathologist annotation, and also keratinocytes are correlated quite well with the pathologist annotation. Yeah, for the other cell types, I'm not sure.
great. Yes.
So have you ever analyzed some treated samples and you can see these different cluster populations of cells, how they respond?
Yeah, for this cohort, we don't have any information about the treatment. Yeah, we just have the fatal and non-fatal cohort.
Hey, let's bring our hands together for Zhao. Oh, no, we've got one more final question.
I just was going to ask about the level of sensitivity or depth, I guess, that you're able to see and what the magnitude of difference had to be to create those clusters and see those differences.
Yeah, so for this kind of technology, actually the signal is quite noisy. So we are not analyze the single cell resolution. So we use a kind of global level to analyze the cell type communities. And in this project, we also have other type of data set, which are more sensitive in terms of identify RNA and proteins. And in our next step, we will integrate multiple technologies to confirm the results. Thank you.
Thank you, Zhao. Okay, I'd like to introduce our next speaker. We've got Shivam Vora, who is doing his PhD with the Gabrielli Lab. Thank you.
Hi, everyone, and thank you for the opportunity to present my work here today. So I'm going to talk about some of the imaging work that I've done in my PhD, specifically looking at high content and live cell imaging of polyploid cells, and just take you all through the kind of data that I've got from these techniques. So just to give a bit of background, we work with aurora kinase B inhibitors, specifically working with an inhibitor that was under development at AstraZeneca called AZD2811. So what is aurora B? It is a protein that is active during mitosis. Specifically, it mediates exit from mitosis. It ensures the correct partitioning of the replicated genome, and essentially it is very important for aurora B to be functioning properly for there to be a successful cytokinesis. So what happens when we inhibit aurora B? So I answered that question with the help of time-lapse microscopy. The way that this experiment was done was after seeding and treatment, the cells are imaged right after treatment, and I've done multiple variations of this experiment for imaging 1 day or imaging up to 5 days, and I've used the Zeiss Axio Observer 7. Basically, this microscope is able to replicate the incubation conditions of the cells, so the cells can be imaged over a long time. And the cells are imaged in a fixed interval, so every 20 to 30 minutes. And then at the end of the imaging, the images are collated to produce a movie. So what kind of data I've received from, I've been able to get from these experiments is to see the outcome of drug treatment, specifically to see the percentage of cells that are failing cytokinesis, and that turned out to be a very important metric for me. To see the amount of time that cells are spending in interphase and mitosis, and I can do that based on the frame numbers from the movies. And also look at the cell fate, and for different for a different kind of research, people might use this to understand the type of cell death that is happening in response to therapies. So let's look at some of these movies that I've got. First, we've got just a control movie where we can see cells entering mitosis. You can see the cells form a dumbbell shape, and then they drop down as two identical daughter cells, exactly what we expect. When we inhibit aurora B, however, the cells are able to enter mitosis successfully after replicating their genome. But just as they're about to divide, they fail and they drop back down as a cell with twice the amount of DNA. And in certain genotypes, such as P53 and RB knockouts, or cancers that have mutations in these tumor suppressors, they undergo multiple rounds of these failed cytokinesis in response to the drug and become polyploid. So polyploidy is basically when a cell has multitudes or like multiple times the DNA of a normal cell. And these cells can get really quite big. So, analyzing this data, unfortunately, I've not been able to find any automated way of analyzing these movies. So I've still, been, doing it manually, but I still, get a lot of insight. So First is a control cell. This is just a representative control cell over 4 days of imaging. And we can see the timely mitosis that takes place in these cells and how we get a huge number of cells at the end of imaging. However, with the inhibitor treatment, all of these timelines are straight lines because the cells fail mitosis 100% of the time in all the experiments that I've done. And we can see in blue, every time the cell undergoes mitosis, and then the limited cell death that happens with this treatment. And from that, I can understand interphase and the time spent in interphase and mitosis, which gives us an idea about what is the response of these cells and how are they responding to the treatment. In different experiments, I've imaged them in a later phase where the cells are much bigger and we see huge delays of these cells in interphase and mitosis, so they're really slowing down. So at the end of our treatment time point, we end up with cells that are really quite big, and like I said, in specific genotypes, they can get really big. And this is what those cells look like. So these are C33A cells. These are cervical cancer cells that have defects in P53 and RB tumor suppressors. And this is just an IF image, which shows DAPI in blue, which stains the nucleus, and then alpha tubulin, which stains the cytoplasm. We can really appreciate the sheer magnitude of these cells. And we call these cells endomitotic polyploid giant cells. And as you can imagine, imaging these cells and being able to analyze, that imaging data is a really important part of my project. So the kind of imaging that I do is high content imaging. So the pipeline of this is that after seeding and treatment, the cells can be fixed, and then we can perform antibody staining on these cells and then image them in high content imaging microscopes such as the InCell Analyzer 6500 HS. I have also used this for live cell imaging, for short-term live cell imaging, and it works really well. Essentially, the way this works is that the microscope is able to, in an automated way, take multiple images, multiple fields in each well. And I work with triplicates or quadruplicates, so I can really image thousands of cells in a really short amount of time. And then finally, I use image analysis software, Cell Profiler, to analyze these cells and then to get single cell data. So like I said, we can do high throughput IF imaging on live or fixed cells. And in some modern systems, we also have automated image processing. This hasn't worked for me because I work with these polyploid cells, and these preset pipelines don't work for them. And also we get single cell data, which is high resolution. And you get a really high number of data points from these images. So you write from information about the size and shape of these cells, to the DNA content, to fluorescent intensity, and really a lot of different parameters can be analyzed. So I want to talk a little bit about the image analysis pipeline, because we've got a lot of people here who've done imaging, and image analysis is a huge part of it. It takes much longer for me to do the analysis than to do the experiment because of the peculiar unique problems that I've faced. So in a traditional pipeline, you'll see the control and the treated cells, while they might be they might die and all of this in different treatments, their morphology doesn't change that much that the same pipeline doesn't work for them. That's a unique problem that I've faced. So the way that this is usually done is that one image analysis pipeline is made to analyze both control and treated, which makes sense, makes the analysis unbiased. But I've had to change the way that this is done while keeping the analysis unbiased, which is to change the inputs that I provide to the program. So I've had to make separate pipelines for both control and treated, so that I'm able to accurately identify the cells. Because if I do it in the traditional way, that analysis was very inaccurate. So by changing the pipeline and then keeping the output parameters the same, I'm able to correctly segment these giant cells. These cells are 2 to 5, 5 to 10 times as big as normal cells, so they cannot be analyzed in the same way. But then still keep the output parameters the same, so I can get unbiased data. So this is what the output from the high content imaging looks like. So this is an image this is an experiment looking at proliferation markers. So we've got Chi67 and EDU intensity. So Chi67 is a marker of proliferation that, what Jordan was talking about, and EDU is a marker of replication. So it shows the number of cells that are undergoing replication at any given point. So the way I do this analysis is after the images are taken, we segment the cells, and then based on the mean fluorescent intensity of these markers, I can designate them as either positive or negative for these proliferation markers, and then I can represent them in this way. Basically here we've got, so these two cell lines, the HT1080 and HCD116, we see huge decreases in Chi67 and EDU after treatment, whereas in cells that are P53 and RB mutant, so these are the C33 and Cascade cell lines, we don't see that decrease. This is another way that I've used high content imaging data. So this is a separate experiment where I was looking at P53 intensity, because that's one of the tumor suppressors that is involved in response to Aurora B inhibition. So we can really, because we get single cell data from high content imaging, every single dot here represents a single cell and its fluorescent intensity. So we can see the, total DNA content versus mean P53 intensity to see how size or DNA content of the cells is correlated to the P53 intensity and how it's getting activated. And also look at replication markers to show that how does the activation of P53 take the cells out of the cell cycle. So P53 is through P21 is responsible for that immediate cell cycle arrest. And in the literature, it was known that when the cells fail mitosis once, P53 activates and it takes the cells out of the cell cycle. But what I've seen through this data is that even though this P53 is activated quite robustly after 24 hours of treatment, where the cells have failed mitosis once, they really don't exit the cell cycle until 48, until another day has passed, so another failed mitosis. And then I went back and I could actually see my imaging data, and that was corroborated that even cells that are meant to arrest in response to this actually take 2 or 3 failed cytokinesis to actually exit the cell cycle. Another way that I've used this is, in collaboration with Matthew Jones's lab at the Fraser Institute, we work with these CDK4/6 and CDK2 reporter systems that were developed in Tobias Meyer's labs in the US. And basically the way that these work is that in response to being, so these reporters, when they're phosphorylated by their targets for CDK2 or CDK4, they get translocated into the cytoplasm, and then we can estimate the kinase activity based on the cytoplasm to nucleus ratio. So we segment the cells and then use the special pipeline for the polyploid cells, and then we're able to estimate the fluorescence intensity of these cells. So we can see here in the wild type, there's a decrease in the CDK2 activity, whereas with the P53 knockout, the CDK2 activity does not go down. And interestingly, even the CDK4 activity does not go down. And finally, some of the recent experiments that I've done with Jennifer Gunter's lab at QUT, I've done some live cell imaging of mitochondria because we are suspecting that these polyploid cells have defective metabolism. And so we've imaged mitochondrial membrane potential and also stained mitochondria passively to see, and get sort of total intensity and mean intensity of these, and that, this is still an ongoing work. And yep, that's it for me. Thank you to all the collaborators in the lab and funding bodies. Thank you. Any questions? There we go.
Thank you. Do we have any questions? Yes.
Beautiful talk. So with your mega cells, you call them that, how are you dealing with the intensity of your different stains? Because you've got pockets or different areas of different intensities within that then would cause your analysis to be really quite problematic as well. So how are you managing that?
Yeah, so, I've dealt with that a lot, because certain stains really do seem to, be quite different But basically, if I keep the protocols for imaging very similar, when I'm staining the cells, I try to keep it as constant as I can, even though, there are features of the polyploid cells that, sometimes they wash off quickly and they are, not so adherent. But essentially, when I'm imaging thousands and thousands of cells, and after segmentation, I get the mean fluorescence intensity that really normalizes those things, and I'm able to compare them directly. Yes.
Great talk, thanks. I was just wondering what happens to your giant cells after you withdraw the drug? Have you ever tried a washout?
Absolutely. Yeah. So, how I mentioned that after the first 2 failed cytokinesis, that's really the tipping point. After basically 48 hours of treatment, if I wash off the drug, the outcome does not change. The cells fail mitosis continuously, even in the absence of the drug. And we've actually, investigated why that happens. And it's because the duplication of the centrosomes is tied to the cell cycle. So as you go further down with multiple rounds of failed cytokinesis, the number of centrosomes actually increase, and then it leads to a multipolar mitosis. And because Aurora B is responsible for the segregation of the chromosomes are not segregated. The DNA is in the middle. It's a huge muddled mass. And then you see these beautiful centrosomes all around the cell, multiple centrosomes. And we've done some mathematical modeling with some collaborators to show that it's very, very unlikely for a single pole to fish out the appropriate complement of chromosomes from that pool. And that's why we never get, proliferation from those giant cells, even in the absence of the drug.
Great, thank you very much for that. Okay, I'd like to introduce our next speaker, Dr. Maria Kojic from the Wainwright Lab. Hi, everyone. I'm Maria, and I'll tell you today more about different types of imaging we're using in our lab to study brain cancer in children. So brain cancer in children is the leading cause of any disease-related death. Very alarming, and there are different types of brain cancer. Predominantly, these are gliomas, medulloblastomas, and ependymomas. But there's so many different molecular subtypes within each of these. There's a high intra and intertumoral heterogeneity, which makes things much more complicated when it comes to treatment. And every year, just in Australia, we have 300 new cases, and the incidence has been increasing basically since ever. So it's very interesting that in the last 30 years, with all what we have achieved in science and technology, we still cannot improve survival rates. So there is an ongoing need to improve the treatment, and we are using various preclinical trials in our lab to address this, but we're also working on improving diagnostic and monitoring measures for these patients, like using liquid biopsy recently. So our research is heavily based on mouse models. We use so-called PDXs, or patient-derived xenografts, patient-derived tumors that we orthotopically inject in the brain of our mice that are immunodeficient mice. But we also use genetically engineered mice to model certain mutations, and syngeneic mouse models, as our lab has recently started using immunotherapies for these mouse models. So obviously we can't work with immunodeficient mice. So for us to run these various preclinical trials for various targeted therapies, and these include not just drugs, but also radiation and immunotherapies, it is very important to be able to image our mice and to monitor the tumor growth. Originally, we used to do that just based on the clinical signs and symptoms in the mouse models, but these are obviously not such an accurate representation of what's exactly going on with tumors, as often these are metastatic tumors, and there is a suppression of brain ventricles, a lot of fluid buildup, head swelling, and you would think it's because of the tumor, but it's actually something else. So, I mean, it's not such a big extent of tumor growth. So what we do in our lab is we use now IVIS imaging. So this is based on bioluminescent imaging, and we do use a lot of MRI as well. I'm sure that most of you are familiarized with the IVIS. So what we do with our tumor models is that we introduce luciferase by just virally treating these lines, the patient-derived xenografts. So we introduced vectors that have GFP and luciferase. We do a lot of fact sorting based on GFP because these are a bit tricky models. PDXs, they don't grow in vitro, so we have to do a lot of in vivo work. And then once luciferase is there, just inject luciferin, use IVIS imaging to monitor the tumor growth. Then, of course, we do a lot of histological analysis, flow cytometry, but also histological analysis. And what we're looking in is that upon using different drugs, different treatment modalities, there is an increase in apoptosis, reduced proliferation, but in some instances, some drugs can also induce differentiation of these tumor cells. So this will be a good outcome for the treatment. And then we analyze this using confocal imaging, and these are massive tumors, so we do a lot of tiling and Z stacking. So this is an example of the current trial we are running in our lab. So you've already heard from, before me about CDK4/6 and its role in the cell cycle. So we use a CDK4/6 inhibitor called palbociclib that has been approved for breast cancer treatment in our mouse models of medulloblastoma. So this is a very aggressive medulloblastoma models, subgroup 3, metastatic. And these are different, you can see here different arms of treatments because we are just using control mice, but also radiation only, palbociclib only, and we're testing a combination treatment with either concurrent palbociclib and radiation or palbociclib following radiation. So to run these preclinical trials, we always have to be aware of the standard of care in the clinics, and that's radiation and chemo, and to combine our treatment with the standard of care, which is why we are trying to test whether palbo in combination with radiation will actually give better results, because we do know that palbo itself is giving remarkable results in these tumors. There is a lot of shrinkage you can see here. And then by just using IVIS imaging in these tumors, you can see that, and based on the just survival rates, that there is indeed a big change when using the combination treatment. So these are just the control group. The mice succumb to tumors in only 20 days. But if using palbociclib or radiation, you do extend the survival. And that's happening even further with the combination treatment. So you even more over doubled the survival of these mice. And this is just as an example of how we monitor the bioluminescent signal. So it's quite accurate. We can also, by using IVIS, track the metastasis. And again, the purple and the orange one will be the combination treatments. You can see that there is a significant reduction in the metastasis of these tumors. So, okay, besides IVIS, we do have to rely on MRI as well for these models. And this is for two different main reasons. One would be that some of the lines we use are not labeled, and they're not labeled often because when you're labeling lines, in some instances, you can also induce some clonal selection and make them more aggressive, and they just do not represent what you see in the clinic anymore. So they're not these tumors that you originally start with. So we sometimes prefer using unlabeled lines. And on the other side, although IVIS is giving you a good indication of the tumor growth and response to the treatment regression, you cannot still accurately determine the volume, the structure, where it's growing, etc. Because we have now a lot of these tumors grow in the cerebellum, but we're working also with gliomas and the tumors that grow in the pons or the cortex as well. So this is an example of medulloblastoma line. again, using Palbo treatment, you can see that this tumor is basically holding. So this is before treatment and after treatment. There is no growth happening during the treatment, but after treatment, it just comes back. So these tumors are not really responding, they're not shrinking, but they do regrow after the treatment. And this is just looking at, these are quite bloody tumors and heavily infiltrating into the brain. And you can observe here, just based on H&E, how they look like. So this is another MRI imaging for another model of medulloblastoma. That's another molecular subgroup where you can see that the tumors actually shrink. So they do respond well, but there is this big head swelling because these are metastatic tumors. So there's a lot of fluid buildup happening. And obviously, upon treatment, we're analyzing these tumors for different markers. This is just one of the examples. You heard a lot about microtubules from Rob today. So this is one of the microtubule stabilizing agents. And it works really great in some of our models. So we observe a lot of reduction in proliferation based on Ki67 expression and increase in apoptosis. This is just cleared caspase 3. Okay, so besides using just contrast MRI, just basic T1, T2 weighted MRI, we also use a contrast MRI, and we use it a lot in our studies of blood-brain barrier. Because as you could see on previous images, tumors do respond, some of them, and they respond in a great way. They almost completely shrink, but they do grow back. And we wanted to also see whether then the whole tumor area is being responsive or not. So we used Gadovist. This is a contrast that we inject just in the tail vein of the mice. And based on these different models, these are 4 different models, you can see that, for instance, in our germline, these are genetically modified mice, that the blood-brain barrier looks quite intact, whilst in other PDXs, the tumors uptake a lot of this agent of Gadovist. So the tumor blood-brain barrier is quite leaky. And this is just quantified here, and this is just measuring the vessel leakiness. So there's a lot of leakiness in these tumors. And then when we histologically analyze them, you can see appreciate some vessels here marked on H&E stains. But when we use specific markers like, PLVAP, which is marker of primitive vasculature, of fenestrated vasculature, so it should not be present in a normal brain vasculature. You can see that it's upregulated in the leaky tumors because these ones are not leaky. And this is just CD31 and GLUT1 reporters, again, marking the vasculature. And if you thoroughly analyze them, you can see nicely the compromised integrity of the vessels. And as I said, upregulation of this fenestrated marker. So then when we spatially analyze these tumors, we could see that these leaky blood vessels are present that express PLVAP. They're present predominantly in the center of the tumor, whilst periphery has quite unleaky or just intact blood-brain barrier. And then further analysis showed that these central areas have an increased expression of HIF1 alpha and other hypoxia-related markers. So basically, this whole mechanism of blood vessel leakiness is hypoxia-driven. And at the end, I just wanted to show you another. So you heard already about spatial transcriptomics, and we did collaboration with Kuan Yuan in this regard as well. And we managed to get really nice imaging using spatial transcriptomics of our models, just based on H&E and MRI. We knew where we could predict where is the mouse tissue, where is the human PDX tissue, and then based on spatial transcriptomics, you can see that
indeed where we expect to have human tissue, we can detect transcripts of human origin only, and then there is this yellow kind of border that's between mixed mouse and human. And we were able to get a very nice resolution of the cells and identify different functional structures and cell clusters in our models. So molecular layer, Purkinje cell layer. So these are specific layers within the cerebellum that we could, just based on the gene expression, validate. And yes, as I said, identify very specific cell types being present in different types of tumors. So this is a very powerful tool for us because, for instance, we know that the edge of the tumor is not responding well. And okay, the blood-brain barrier is really compromised there, but let's say these are palbociclib treated mice, and palbociclib does cross blood-brain barrier very well. So why then the cells do not respond at the edge? And then we could just spatially analyze these mice, these tumors, and see that there's a lot of astroglia, microglia enriched pockets there, and proliferating cells in those specific areas. So as I said, these are very heterogeneous tumors, so it's very important for us to know spatially exactly what's going on. And this is just our lab. We're here at the Fraser Institute, and happy to talk later if we're now have to stop because of the time, if you have any questions. Thank you, Maria. We might just have the one question. So no one's starving, are they? We can wait. Close to lunch.
Thank you. You have mentioned that the trillion brain has the intra-patient heterogeneity. So is it possible to detect this heterogeneity through MRI?
Sorry?
Is it possible to detect the heterogeneity through MRI screening?
Through MRI. Yeah, it's a good question. Well, not really. So if you would use the contrast MRI, you would obviously see where is the blood-brain barrier leakiness that you can definitely nicely capture. And indeed, those pockets that are not leaky, where there is an intact blood-brain barrier, you will see that the tumor is coming back there. But that's all you can see. So no, you can't. These are molecular changes. So it will be based on spatial transcriptomics, you would be able to detect a lot of it. Yep. Thank you.
Thank you, Maria. That was amazing too. I've been really impressed with the speaker lineup today. And we're coming to our last speaker today, Dr. Alexis Bonfer-Mello, who is a postdoc in the Jones Lab at Fraser Institute. Thank you, Alexis.
Oh, I think we are missing a few slides in here. Okay. Okay. That's fine. Okay.
Do you want to have a look at it?
Yeah, we are missing 2 slides. Yeah, in the beginning. That's fine. That's fine. That's right. We are 5 minutes from lunch.
Which one was yours?
The second one. Yeah. Last week, right. Okay. Yeah, thanks a lot, Shannon. Thanks for the other speakers for rearranging your schedule to accommodate my personal things. And I was voiceless during the weekend. Hopefully I go back and let's see what I have to present to you. So in our lab, today I'll just show you briefly how we use high content imaging, so quantitative image-based cytometry to study cell cycle in our lab. So I'm postdoc with Matthew Jones here in Fraser Institute. So the cell cycle is really regulated by pairs of CDKs that are cyclin dependent kinases and their pairs, the cyclins. So the cell cycle is regulated by these pairs of proteins that activate and lead cells into the cell cycle. So for example, here we have CDK4/6 that Maria just talked about. They are the main ones to lead cells from G1 into the S phase. We also have CDK2 bound to cyclin E, and these are the two main kinases progressing cells from G1 into the S phase. And in the S phase start for DNA replication is mainly led by two kinases. So CDK2, one is bound to cyclin E and cyclin A, but also DDK. So DDK is our favorite protein in the lab. It's CDC7 bound to its cofactor DBF4. And these two kinases, they phosphorylate the helicases that are the proteins that unwind the DNA for the start of S phase. And they are the key proteins for DNA replication, DNA synthesis. Their activity can also be regulated by what we call S phase checkpoint, ATR, CHEK1, and WEE1. They are kinases that inhibit this activity, and they are the proteins that take place when we have replication stress or DNA damage responses. So what happens when we inhibit cells, CDC7 and in cells? So Matthew had his postdoc in the US, in York University, and he investigated that. So when we inhibit CDC7, we still have cells entering S phase. So these are flow analysis in which we have classical cell cycle profile with G1 cells with low quantities of DNA, and this is BRDU incorporation. This is a thymidine analog that stain cells that just entered into the cell cycle. So when they enter in S phase, they get BRDU positive, and when they finish, they go BRDU negative into G2. So this is a normal profile for control cells. And when we inhibit CD7, we still have cells entering S phase, but they fail to finish S phase. And in the end, you have a big late S phase arrest when we inhibit CDC7. So Matthew just saw that despite CDC7 is also responsible to trigger the helicase activation for DNA start, CDC7 also has a role for S phase termination and also a role that I will not go into the details for the S phase checkpoint. So moving here to Australia, I joined his lab, and my main focus, or the main focus of my work, was to understand how is CDC7 and CDK co-regulating S phase entry in cells. So we know quite a few kinases that are accessory for CDK in the mitosis checkpoint. And now our question is if DDK, CDC7 is one of the accessory kinases regulating CDK2 entry into S phase. So we did that using kinase translocation reporters. So you just saw in Shiv's presentation that we can use these kinase translocation reporters. In this case, this is a CDK2 activity reporter. When we have CDK2 active, this reporter is translocated to the nucleus. And we got this reporter in the lab, and we did some live cell imaging. So this is the reporter, and we track the cells. This is PCNA that shows individual factories where we have DNA replication, and this is CDT1. That is one of the proteins that are regulated during S phase. And what we can appreciate is the increase of CDK2 activity during the cell cycle, and at the start of S phase, that's when we have fully degradation of CDT1, is when we have, we can check CDK2 activity at this point, and it keeps going up till the end of the cell cycle. So this is S phase. Once S phase finishes, CDT1 comes back, and we have the highest amount of CDK2 activity, and the cells divide. So the main question was, okay, how is CDK2 activity when we inhibit CDC7? Because the hypothesis is that we have these two proteins cooperating to get cells into the S phase. And in normal circumstances, so we track a bunch of cells in a microscope and we align their CDK2 activity and PEP profiles. And these dots are at the moment of S phase start. So in normal circumstances, that's what we have. At a certain level of CDK2 activity, cells enter the S phase and they progress. When we inhibit for the CDC7, what we have that at that stage, cells still don't enter the S phase. They have to reach a new threshold of activity. They have to work harder to get into activity level that they enter S phase. And what we can do is to get these cells and evaluate CDK2 activity of the entry of S phase. So in normal circumstances, we have this level, but when we inhibit CD7, the CDK2 activity has to be higher. So really suggesting that CDC7 and CDK2, they work pretty much in same mechanism to get cells into S phase. When you remove one, the other has to work harder. And yeah. So this is live cell imaging, but you can really not get many data points with live cell imaging. So what we had to do was to go to high content imaging. And one of the problems with that is with flow. You cannot track these reporters because they rely on the nuclear cytoplasm translocation. So we went to IF, and this is a typical staining, but this is coming from this kind of acquisition. So large fields of view acquisitions. And we do these experiments in 96 well plates, as Shiv mentioned. So in the beginning, so we did most of our work using the in-cell analyzer that we used to have here in level 6 from Derek Richards. Now it's located in level 5. It's still available. It was broken, but it was fixed. It was a very dark 6 months that we had doing experiments in none other systems. But hopefully Mark Scott's here and can confirm. So we'll have the operator in the facility. There was a huge effort from Matthew to get this machine in here, and Mark Scott, of course, helped with that. So these are the systems. So we do our immunofluorescence in these plates, put in these machines, and do acquisitions of thousands of cells. So in good experiments, you can get like 500,000 or 1 million cells from these plates. And the really powerful step of that, just like Shiv said, is to do the image analysis. So in the lab, we have a more user-friendly one that goes through Fiji, start this machine learning segmentation and cell profiler, but we also have some hardcore one developed by David that relies in a lot of coding by Python and much better segmentation by cell pose. Both of them generate massive spreadsheets that GraphPad doesn't like, neither Excel. So we have to put that into RStudio. And the beauty of this technique is, as Shiv mentioned, is to get subcellular localization that's crucial for these kinase translocation reporters, is to evaluate subpopulations of cells and also molecular and phenotypic phenotypes. So today I will just really try to go very briefly in few stories that we have in the lab. So the power of this technique is that in normal circumstances, in all days, for you to evaluate the kinase activity of these proteins, you have to synchronize the cells with drugs with serum starvation, and then that can potentially cause artifacts. But now, with high content imaging, you don't need to synchronize the cells. You just get the cells in unsynchronous way and interrogate them. So this is the classical DAPIDU plot that you just saw that we have with flow. And this is in normal circumstances. So this is the G1 population. This is S phase, and this is G1. And the question in here is, so how does CD7 activity looks like with high content imaging. And this is the activity, the phosphorylation of one of CD7 substrates. And what we can see is there is increase of CD7 activity throughout the cell cycle. And when we inhibit CD7, of course, you have the decrease of this substrate. We can check these intensities as a factor of DAPI, and we can see the decrease of that in the whole population. And the beauty of that is to evaluate the subpopulation. So we can gate the subpopulations and interrogate how the phosphorylation, the activity of these kinases changes in the cell cycle. So this is just an example. So we can already appreciate that CD7 activity, although that's a kinase that regulates DNA replication, it already starts at G1. So it already phosphorylates its substrates as G1, and then the phosphorylation ramps up. And when you have thousands of cells, what we do usually, we get the means of these experiments to do proper stats. What about CDK2 activity? So what we have for CDK2 activity, when we inhibit CD7, we can reproduce what we just saw in live cell imaging. We see increased CDK2 activity. And when we gate these populations, we can see there is a higher CDK2 activity when we inhibit CDC7, but most important than that is that these cells, they are indeed stuck in G1. So in normal circumstances, cells with high CDK2 activity, they already been in S phase, but in the absence of CDC7 activity, they are stuck in G1, very similar to the plot that I just showed us. These cells are struggling to get from G1 to S. And so really corroborating that CD7 is cooperative with CDK2 to push cells into cell cycle, into S phase. The second little story is showing how we can further evaluate this synergy. So we did these experiments with local student in the lab. So the beauty of doing high content imaging is that you can fully explore a bunch of conditions. And in this case, we use combination treatments to inhibit CDC7 and CDK2 and evaluate what is the profile in the cell cycle. So this is CDK2. We have a decrease in S phase in DNA incorporation, and this is CD7, we have this decrease. And when we measure EDU incorporation, we can really appreciate at certain levels of CDK2 inhibition, we have a sensitization of these responses with CDC7 inhibition increase. We can adapt these measurements for synergy analysis and really depict the cooperation between these two kinases with this kind of plot. So this is inhibition of EDU incorporation, and this is the synergy score that we usually have. So this is for RP1 cells. They are immortalized but no transformed cell lines, but we also have some data with cancer cells. We truly skip the two other stories just for the matter of time. So we can also do some, David is doing quite cool analysis and exploring cyclin E overexpression in CDK4/6 resistant cells, doing a bunch of titering of these drugs. CDC7 inhibition induces gamma H2AX, that is a DNA damage response marker. And you can really appreciate these damage populations rising up with the concentration. So we can really interrogate this phenomena with that. And finally, so we can also study blood cancers. So we can also study suspension cells in high content imaging. So when you do live imaging with them, they just don't adhere. But if you coat and spin them, they can remain attached in there. And you can do this analysis in blood cancers as well. So this is MV411. So this is AML cell line that we are studying in the lab. I'll just not go too much into detail we have the lunchtime, you can come over and have a chat about that. Yeah. And the irony is that when I talked about high content imaging to Gustavo that use a lot of flow, he said, yeah, but you cannot put like 20 antibodies or anything like that. But the reviews just ask them to get imaging confirmation of their facts, and here I was to help them. Good. So thanks a lot for your attention, and sorry for going over the time. Thanks, Shannon. Yeah.
Do we have any questions? We'll have one and then we'll. Right up the back there, we've got a winner.
Might come and hit you up at lunchtime. What have you used as your coating?
Sorry?
Your coating for getting the cells to stay.
Oh, how do so I tested fibronectin, polylysin D, and a bunch of centrifugation steps. In the end, I'm using polylysin D in coating 1 hour. I can give you the protocol.
Yeah, I really want to hear about any differences that you saw in your staining over that. I'm going to come and find you, I think.
Yeah, sure.
Okay, we might finish up. Thank you very much, Alexis. Round of applause. I think we can all agree that the EMCRs are doing a fantastic job in this field of cancer imaging. And you can see we've gone through a range of cancer imaging modalities, looking at the cellular level, the spatial level, and how that translates to tumours. So I hope you enjoyed the talks today. Lunch is being served, so you're welcome to come and find our speakers and ask them any further questions. Thank you for your attendance.